Substances such as nutrients, gases, and waste are constantly moving into and out of cells. When the concentration of a substance is higher on one side of the cell's permeable barrier, its molecules use osmosis or diffusion to move through the barrier without the cell using any energy. This process is called passive transport. When the molecules of a substance need to move from an area of low concentration to one of high concentration, they use specialized proteins in the cell membrane. This process expends energy and is called active transport.
Cellular respiration is comprised of three main steps. It begins when glycolysis breaks down a 6-carbon glucose molecule, producing 2 3-carbon pyruvic acid molecules and a net gain of 2 ATP molecules, which the body uses as cellular energy. High-energy electrons are released. Each 3-carbon pyruvic acid molecule then enters the mitochondria as part of the second step, the citric acid cycle. 2 more ATP are created and carbon dioxide is released as a waste product. High-energy electrons are again released. The third and final step is the use of high-energy electrons in the electron transport chain, located in the mitochondrial membrane. It is during this phase that most of the ATP molecules are created. The previously released electrons interact with a series of enzymes that store the energy in the electrons in ATP molecules. The 3 steps of cellular respiration can produce up to 38 ATP molecules from one glucose molecule. All cellular gene expression and protein building occurs through the 2 principal steps called transcription and translation. In transcription, DNA's information is encoded or transcribed into messenger RNA or mRNA. The process begins when RNA polymerase opens the double helix of DNA, like the slider on a zipper. It unwinds the DNA double helix and pries the 2 DNA strands apart. As the RNA polymerase continues down the length of the DNA, several actions take place. It reads one strand of the DNA and transcribes the DNA's information. It creates an mRNA version of the DNA, or transcript. The mRNA transcript is a complementary copy of the DNA sequence. When the transcription ends and the mRNA template is complete, it separates from the DNA template and exits the nucleus through pores. The template enters the cytoplasm on its way to the next step in the process of polypeptide synthesis, translation. Translation begins when the mRNA enters the cytoplasm and a small ribosomal subunit attaches itself to the end of the mRNA strand. At the same time, a transfer RNA, tRNA, molecule recognizes and binds to the start codon on the mRNA strand. tRNA recognizes the mRNA codon through its anticodon, which has a sequence complementary to the mRNA codon. It is the tRNA molecules that carry the amino acid building blocks of the protein. Next, a large ribosomal subunit joins and forms the fully functional ribosome that will join the amino acids on the tRNAs to synthesize the protein. When the amino acids of the two tRNA molecules are joined, the tRNA in the first position is released to get a new amino acid. The growing protein is attached to the tRNA in the second position. The mRNA is then pulled through the ribosome so that the tRNA with the protein chain is now in the first position. Now, a new tRNA with the proper anticodon can come into the ribosome at the open position. This pattern continues as tRNA after tRNA bind to the mRNA codons and their amino acids are added to the growing protein chain. Toward the end of the mRNA sequence, the ribosome gets to a stop codon, which does not code for any amino acid. This signals the end of the protein and that the translation process is complete. The ribosome then releases the finished protein from the tRNA and detaches from the mRNA as the two ribosomal subunits separate. The protein will then be further modified in other cellular structures or directly used by the body.